All right, number 14. In an experiment, cells were isolated from an aquatic plant and suspended in pond water, a sucrose sugar solution, or distilled water. All of the cells were then viewed under a microscope. Compared with the cell in the pond water, the cell in the sugar solution appeared shriveled, and the cell in the distilled water appeared inflated. The results of the experiment are represented in Figure 1. Which of the following statements best explains the observations represented in Figure 1? So first I want to add some terms to this. The cell that's in the sugar solution, what kind of word could you use to describe that? Yes, plasmolized. I don't know where I'm going to fit this. Plasmolized. It's very thin for some reason. How about the cell in distilled water? How could you describe that one? What's the other word that you could use? It begins with a T. So close. Turgor. Such a weird word, isn't it? Turgor. Yeah, you got the hyper and the hypo, but you at the end. So another way to say turgor is that it's turgid. I like turgid better. Turgor just sounds really very strange to me. I think it does. Plasmolized is still the best word, though. Yeah. All right, so what can we eliminate? What can we do with these here? Okay. Why are we eliminating A and B? Excellent. So the sucrose is not what's moving. It's the water that's moving in these particular cells. Good. So now these two have the movement of water. One is out of the cell in the sugar solution, and one is into the cell in the sugar solution. So if we look at that one, what happened to that particular cell? Where did the water go? It went out of it, right? So our answer is C. You don't even have to look at the ending part because we know that part is correct, and we only have those two options left. So one of them has to be the right one. Number 15. Changing the shape or morphology of the mitochondrial inner membrane can change the efficiency of the mitochondrial function. Which of the following outcomes would most likely result from changing the shape from highly interfolded to a smooth, flat surface? So our mitochondria has that folded inner membrane, and if we took that and just made it a smooth, flat membrane, what would happen? Yep, this one has less surface area, right? That's the whole point of the folds, is to increase the surface area to make it more efficient. So what do we want to do for an answer here, James? Right. These two talk about it becoming more efficient. Yep, because we're going to decrease that surface area. So D is the correct answer. Number 16, the figure shows a process by which a cell might absorb food from its surrounding environment and break it down for use as a source of energy and matter. The process involves lysosomes, which are membrane-bound organelles that contain hydrolytic enzymes. Activation of the hydrolytic enzymes requires an acidic pH, and lysosomes maintain an internal acidic pH by using ion pumps. Which of the following outcomes will most likely result from a loss of ion pump function? So before we even look at the options, what do you think is going to happen? If we lose the ion pump function, what's going to happen to the internal environment of the lysosome, it's going to become less acidic, right? Sometimes it's good to just think about it before you even look at the options and see if you have a little bit of an idea. So what do you want to get rid of?
Oh, no, yeah, sorry.
So if it becomes less acidic, that means that the pH is going to increase.
Oh, yeah, thanks.
No, that's okay. That's right. The smaller numbers on the pH scale are more acidic, and then the base is increasing. So we're going to get rid of the ones that say that the pH will decrease. So we have B and D that both talk about the pH increasing. Is that increase going to prevent the activation of the enzymes, or will it activate those enzymes? It's going to prevent it, right? It's going to stop those enzymes from working because those enzymes had a very specific range of pH that they could work at. So if we move outside of that range, what's going to happen to those enzymes? The enzymes will be denatured, so they won't function anymore. We're not going to dwell on number 17 because I don't like the question and I don't like the answer and I don't think it's a very good one. So we're going to skip number 17, although the correct answer according to the College Board was C. Number 18, researchers claimed that a particular organelle originated from a free-living prokaryotic cell that was engulfed by a larger cell. Which of the following provides evidence to best support the researcher's claim? If the organelle originated from a free-living prokaryotic cell that was engulfed by a larger cell, which one of those matches the best? No. It is C, that the organelle has a double membrane, right? If it was an organelle that was engulfed by another cell, it's going to have two membranes around it. So having that double membrane is the correct answer. There are a lot of organelles that have a phospholipid membrane around them, so that's not unique to the organelles that they're looking at. What organelles are they looking at that this happens to be? What are the two organelles that provide evidence of the endosymbiotic theory? No, it's a plant one. The chloroplast. Yep, so those are both double membrane organelles. Number 19, which of the following statements best supports the claim that certain organelles within eukaryotic cells evolved from free-living prokaryotic cells? This is all about the endosymbiotic theory again. It is. We just talked about how it's the mitochondria and the chloroplast are the two organelles that have these particular features. What are those special features? We did talk about this in the notes. What do they have unique to them that the other organelles do not have? One of them's the answer from the last question. They have a double membrane. So they have two layers of membrane. They also have their own enzymes. That means they have their own ribosomes to make those enzymes. And that means they have to have their own genetic material to give the code to the ribosomes to be able to make those enzymes. So D is our correct answer. The other feature of these, just to add to this, is that they can self-replicate. Number 20, the endosymbiotic theory proposes a model for the evolution of mitochondria. According to the model, an ancestral eukaryote engulfed a small, free-living prokaryotic organism. The engulfed prokaryote then formed an endosymbiotic relationship with the eukaryotic host. Which of the following observations best supports the model? So this goes right along with this list that we just made for number 19. So if you look at these options here, are there any that you can get rid of or any that you can say this is it? Get rid of A. Okay. Yep, that really doesn't mean anything about the endosymbiotic theory, right? What about B? The mitochondria. Those are not the correct organelles, right? It's the mitochondria and the chloroplast. So C or D? Okay. Why would you go with C?
Also, I think C because they need energy. You're talking about the enzyme. Yeah.
Yep, the mitochondria's job is to actually make ATP. Yep, so that is the correct answer right there. And if you think about it, if we're trying to prove this endosymbiotic relationship, we want to show that there's some similarity between them. Okay? So the mitochondria and some prokaryotes share a similar process to make ATP.